Hello dear students, in today's class we are going to discuss about the spermatogenesis. To understand the spermatogenesis, we have to start with the gametogenesis. Let us see what is gametogenesis. Gametogenesis is nothing but it's a formation of gametes within the gonads or primary sex organs. The gonads or primary sex organs in male are testes, in female they are ovaries. First of all, let us write the definition. It's the formation of gametes within the gonads or primary sex organs. They are testes in male and ovaries in female. So what we have studied? We studied what is the meaning of gametogenesis. Gameto means gametes, genesis means synthesis or formation. And this formation of gametes takes place within the gonads or primary sex organs. Primary sex organs in male are testes, in female ovaries. There are two types in gametogenesis. Let us see what are those. There are two types. One is Spermatogenesis. Another one is Oogenesis. Spermatogenesis and Oogenesis. Spermatogenesis is the formation of male gamete or sperm within the testes. It's the formation of male gametes or sperms within the testes. Oogenesis is the formation of egg or ovum, or you may call it female gamete within the ovary. Within the ovary. But in today's class, we are going to study only spermatogenesis. In the next class, we will discuss about the oogenesis. Let us discuss in detail about the spermatogenesis. Spermatogenesis. So already we know the meaning or the definition of spermatogenesis, which means formation of male gamete or sperms within the testes. This whole spermatogenesis process has divided into two stages. So the first stage, there are two stages. The first stage is formation of spermatids from germ cells. This one is called the first stage. The second stage is we call the Spermiogenesis. Spermiogenesis. So these are the two stages where the 50% or more than 50% of spermatogenesis process takes place in first step, that we call the formation of spermatids from the germ cells, and remaining part of the formation of functional sperm process takes place during the spermiogenesis. What exactly happens during spermiogenesis means here the non-functional, inactive, non-motile spermatids are transforming into the spermatids are transforming into the motile functional active sperms or spermatozoa. The non-functional, inactive, non-motile spermatids are transforming into the sperms which are active, functional and also motile. To take these changes, there are a lot of changes takes place, and they are morphological and physiological changes which help the spermatids to transform into the sperms. What are those changes? We'll discuss later on while we are studying spermiogenesis. So now what is the definition of spermiogenesis? Spermiogenesis is nothing but it's the transformation of spermatids into the sperms, which we call the second part or second stage of spermatogenesis. But in first stage, what happens? The germ cells undergo sequential changes and they form the spermatids. Germ cells to the spermatids is considered as a first part or first stage of spermatogenesis, and spermatids to the sperms formation is called the second stage. Now let us discuss first stage of spermatogenesis. The first stage of spermatogenesis is the formation of spermatids from the germ cells, which further divided into 3 stages. To complete this whole process, it requires 3 stages. There are 3 stages or 3 phases are there, and that we can say the first one is multiplication phase. Multiplication phase. Second stage we call the growth phase. Second phase is growth phase. And third phase is called the maturation phase. Maturation phase. So these 3 stages are required to complete the first stage process. So the formation of spermatids from germ cells, this process is happening or completing only after these 3 stages. Let us see how all these phases are taking place during spermatogenesis. So spermatogenesis process takes place in the seminiferous tubules of testes. Hence we call the seminiferous tubules are the structural and functional unit of testes. The wall of the seminiferous tubule has made by a germinal epithelial layer. We already studied in our previous class, while we are studying the microscopic structure of the seminiferous tubule or testis, we have discussed everything. But one more time I want to repeat the same thing because to understand very easily the whole process. So spermatogenesis takes place in the seminiferous tubules of testis. And these seminiferous tubules are called the structural and functional unit of testis. And seminiferous tubules are lined by germinal epithelium. Germinal epithelium layer. So this germinal epithelial layer is made by 2 types of cells. So this layer is made by 2 types of cells. One is germ cells. Germ cells. Another type of cells are called the Sertoli cells. The germinal epithelium is said to be the wall of the outermost layer of the seminiferous tubules, and which is made by 2 types of cells. First type of cells we call the germ cells, and second type of cells we call the Sertoli cells. For example, I am going to draw the diagram also because you can understand very easily. This is the germinal epithelial layer and these cells are called the germ cells, and here and there some conical shaped cells are there. These conical shaped cells are called the Sertoli cells. These are the germ cells and these are called the Sertoli cells. The germ cells undergo many sequential stages and at the end of the process they form the sperms. Means these germs are involved in the formation of sperms. But Sertoli cells are there along with germ cells, but they are not involving in the process of spermatogenesis. But they help the spermatogenesis process by providing nutrition for developing sperms. Means they are contributing in spermatogenesis only by providing nutrition to the sperm or spermatozoa. They are not producing any sperms, but sperm production takes place by these germ cells. So now we will start with the three phases. Already we have mentioned the three phases are required to complete the spermatogenesis process. The first phase is called the multiplication phase. Multiplication phase. So multiplication means it's a process where a cell is undergo division, like mitotic division, which results the group of cells. Here the only germ cells are involving in the multiplication phase. Sertoli cells are not multiplying. They are not undergoing any type of cell division. So keep it in mind. So I am going to take the one cell for example, because the same process is observed in every cell. So by taking one cell or one germ cells, I am going to explain you the process of spermatogenesis or the first phase of multiplication phase. This is one germ cell of the germinal epithelium which undergo repeated mitotic division. And produce many daughter cells. And it produces the many daughter cells which are diploid. The germ cell is also diploid, and by the mitotic division which produce the many cells which are also diploid. And these diploid daughter cells produced from germ cells by the division mitosis, and these cells are called the spermatogonial cells. These cells are called the spermatogonial cells. But these spermatogonial cells we are dividing into two groups. The first group of spermatogonial cells we call the type A spermatogonia. They are called the type A spermatogonia, or we may also call the stem cells. Means these cells are dividing by mitotic division and forms the many cells, and they are again spermatogonia only. But they are never undergo further stages. They never enter any further stages. So growth phase, maturation phase, these are not come crossing in this type of cells. These are only helpful for the formation of many more spermatogonial cells. So they are producing only spermatogonial cells. Cells by mitotic divisions. These cells are not entering into the next stages of spermatogenesis process. And the second type of cells we call the type B cells. Type B spermatogonial cells. Type B spermatogonial cells. And these cells are entering the next stages of spermatogenesis. The second stage is growth phase. So this whole process we call as a multiplication phase. So what are the things happening in multiplication phase? So the germ cells, most of the germ cells, because I am taking only one germ cell for our convenience. So most of the cells undergo mitotic division, but I am taking one cell, and this germ cell undergo repeated mitotic division and forms the group of cells. And among this group of cells, half of the group of cells are considered as a type A spermatogonial cells or stem cells, which are further undergoing mitotic division and produce the again spermatogonial cells only. They are not entering any another or next stage of the spermatogenesis. They are not entering the growth period or maturation period. They are producing only spermatogonial cells. And another group of cells of spermatogonia are called as a type B spermatogonial cells, and these are undergoing the second stage and third stage of spermatogenesis, and they are responsible for the formation of spermatids and later sperms. And this whole process we call the multiplication phase. The second phase is called the growth phase. Growth phase. And this growth phase is seen only in the cells of type B spermatogonial cells. So type B spermatogonial cells undergo the second phase, that is growth phase. These cells enlarge in their size due to growth. And this growth or enlargement or increase in the size occurs due to the accumulation of nutrition. Here one thing we should think or understand very clearly, that is they only increase in the size. There is no change in the chromosome number. So chromosome number of these cells and chromosome number of these cells is always same. And these cells are also diploid and these cells are also diploid. The only thing which takes place or change which takes place during this phase is increase in the size of the cells, and that increase is due to the accumulation of nutrition. And the nutrient materials are accumulating in the cells, the size of the cell increases, and that we call as a growth of the cell. And this is said to be second phase of spermatogenesis. And these cells are now we call as a primary spermatocytes. They are called the primary spermatocytes, which are diploid. So the next stage we call as a maturation phase, during which meiosis takes place. Meiosis is the main division which takes place during maturation phase. Here the primary spermatocytes are said to be the parental cell, because these cells are undergoing, I am taking only one cell first. These are undergoing the first meiotic division. The first meiotic division results the two daughter cells. Two daughter cells from one cell, two daughter cells from another cell. And these daughter cells which are formed by the primary spermatocytes are called the secondary spermatocytes. They are called the secondary spermatocytes. But here there are many points, very important things we have to understand. What are those things? Primary spermatocytes are diploid, but secondary spermatocytes are haploid. These are haploid cells, because the first meiotic division we call as a reductional division. It's called as a reductional division. Reductional division means the chromosome number of parental cell is going to be reduced to its half number in their daughter cells. Here the secondary spermatocytes are the daughter cells of primary spermatocytes. The chromosome number of this cell are 46, but the chromosome number of these cells are only 23. Means they have diploid number of chromosomes, but these cells have the haploid number of chromosomes. This is the thing which happens during first meiosis. And second thing is, the genetic recombination takes place in between the homologous chromosomes. The homologous chromosome undergo crossing over, which results the new genetic recombination, which is different from the parental cell. Means genetically, the daughter cells produced from these parental cells are different, because the homologous chromosome undergo crossing over process and that results in new genetic recombination in the daughter cells. So these are the two main things which are happening in this period. The first thing is chromosome number of these cells are reducing to the half. Means these cells have 46 now chromosome, means daughter cells have the 23 chromosomes. Each cell has 23. This cell has 23, this cell has 23, means half of the parental cell. And another thing is, very important thing is, the new genetic combination is observed in the daughter cell. Hence this meiotic division is called the reductional division. Next or second meiotic division. The second meiotic division takes place in these two daughter cells, and they produce 4 cells, two cells from each cell. So totally 4 daughter cells are formed by these two parental cells, and these daughter cells are called the spermatids. And spermatids, and this division is called the meiotic 2. And all cells are haploid. All these cells are haploid. And hence the meiotic 2 division is called the equational division. Means the chromosome number of daughter cell is same as that of their parental cell. There is no change in chromosome number takes place between the parental cell and daughter cells. So the same number of chromosomes are retaining in the daughter cells. Hence this division is called the equational division, or which is also called the similar to the mitotic division. And this process, or up to this process or spermatid formation, we call as the first stage of spermatogenesis. Here, from starting from germ cells, spermatid formation is completed. And this whole process we call as a first stage. But it is not the complete of spermatogenesis, because these spermatids are non-motile, non-functional, and they are inactive. So they should become motile, functional, and active. And all these things are taking place in the second stage of spermatogenesis, that we call the spermiogenesis. So the next stage is spermiogenesis. So already we have discussed what is the meaning of spermiogenesis. Is the transformation of spermatids to the active or functional sperms. So this is we may call the single step process. But what are the changes which are taking place? So these changes are morphological changes, and they may also physiological changes. Means due to morphological and physiological changes of spermatids help to form the functional sperms. Let us discuss what are the changes which are helping to transform a spermatid into the sperm. Changes which takes place in spermatids to form sperm. The first change is condensation of nucleus or genetic material. Condensation of nucleus. Nucleus is condensing, become compact, or a genetic material is become homogeneously spreaded mass-like structure. This is the first change. Second change is formation of axial filament. Axial filament. The axial filament present in the tail region, starting from the neck region, which produce from the one of the centriole, and it is present inside the tail region, which give the strength to the tail of the sperm. And third change is the change in mitochondria and centrioles. So the mitochondria are arranging around the axial filament spiral. So these mitochondria, they are arranging spirally around the axial filament, and that structure we call the Nebenker. That we call the Nebenker. So what is the Nebenker? It's the arrangement of mitochondria around the axial filament. They are arranging spirally around the axial filament. That structure is called the Nebenker. And the centrioles are locating in the neck region. Just below the head, the neck region is there. There is a small constricted region that we call the neck. Within that neck, there are two centrioles are locating. One is proximal and the other one is distal. And next is lack of cytoplasma. Lack of cytoplasma. There is a lot of reduction in the content of cytoplasma. So the amount of cytoplasma is reducing from the cell or from the spermatid, and this cytoplasma is restricted only to the tail region. So hence the sperm contributing only its nuclear material in fertilization, but cytoplasma is not entering. So the sperm cytoplasma is not taking part in the fertilization. And these are the changes which are. And one more thing is the formation of acrosome. This is very important. Formation of acrosome. Formation of acrosome. So this acrosome is derived from the Golgi body. It is derived from the Golgi body of spermatid. And Golgi body to acrosome. And this acrosome is present on the head of the sperm, and which is containing hydrolytic enzymes. This acrosome is made by hydrolytic enzymes. And these enzymes are helpful to dissolving all the membranes of egg during fertilization. So these are some of the morphological and physiological changes taking place during spermiogenesis, and that may help to convert a non-functional, inactive, non-motile spermatid to become active, functional, motile sperm. So we are going to label and draw all these changes while we draw the completely mature sperm structure. So next process we call the spermiation. Spermiation. This is another technical term. Spermiation is nothing but a release of completely mature sperms from the head of the Sertoli cells. During the course of development of sperms, these sperms are attaching to the head of the Sertoli cells. So after the complete development of these sperms, they are releasing in the lumen of the seminiferous tubules, and that process is called the spermiation. Spermiation means release of completely developed sperms from the head of the Sertoli cells to the lumen of the seminiferous tubules. So there are three technical words we have come across during this spermatogenesis process. What are those? Don't get confused and don't think they all are similar. The three words, technical words which I have to remember, they are: one is spermatogenesis, spermiogenesis and spermiation. Spermatogenesis is the whole or complete process which starts from the germ cell up to the formation of sperm. The whole process is called the spermatogenesis, and it takes place in the seminiferous tubules. And spermiogenesis is the process where the spermatids are transforming into the sperms, and this is because of all these changes which takes place. Because of all these changes, non-functional, non-motile, inactive spermatids are transforming into active, functional, motile sperms. And the last is spermiation. Spermiation is the release of developed or completely mature sperms into the the lumen of seminiferous tubules from the head of the Sertoli cells. So these three words are very important. And all this process completes, or it takes 74 days. In human being, to complete the whole spermatogenesis process, it needs 74 days. So here I am going to finish this process, spermatogenesis. So in the next class, we will continue the same topic next segment.